This protocol facilitates the use of human pluripotent stem cell-derived neurons for high-content screening applications. It is particularly well suited for assaying synapses, which in human neurons require lengthy culture periods. We demonstrate the replating of neurons from large format dishes into HCS-compatible multi-wells in a way that preserves their viability. Counterintuitively, we show that extending protease incubation prior to resuspending and replating yields improved neuronal survival.
Human pluripotent stem cell-derived neurons are increasingly relevant in the areas of basic research, drug development, and regenerative medicine. This gentle trituration method can be used to resuspend any large monolayer of cells having a thick network of processes. In addition to human hPSCs, it can be used to replate culture of primary neurons or to resuspend them for FAC sorting or single-cell sequencing.
It is important to follow the protocol steps carefully and frequently monitor the protease-mediated detachment of the neurons from the plate. The optimal incubation time may vary depending on the culture. Visual demonstration enables even inexperienced investigators to reproduce this procedure.
Start by gently rinsing the plate of differentiated neurons with PBS. Disperse the PBS down the wall of the plate and not directly onto the cells to avoid disrupting them. Aspirate the PBS from the edge of the dish while tipping it, taking care not to touch the cells. Apply at least 1 milliliter of proteolytic enzyme per 10 centimeter plate and return the cells to